All right, we'll go ahead and get started. Want to welcome everyone to session 3 of this year's Brown Bagger. It has been mentioned before, but in case this is your first time, we have kind of switched over in terms of our umbrella organization that we work under. It's always been the NBCEC, but since that has not been funded for quite some time, we decided, and we have our other organization, eBEEF.org, going on. We thought it would be more appropriate to place the Brown Bagger under that umbrella. That's where you'll be able to find all of the recordings and things like that. So eBEEF.org, if you haven't checked it out, please go and check out the website. All right. My name's Darrh Bullock. As you can see, Bob's picture of me is from a couple of years ago, so things have changed just a little bit, but I'll be your host today. I'm at the University of Kentucky and primarily do extension here at UK. And again, there's our website where we would like for you guys to go. There's a lot of information on the website. It's not just housing the brown baggers, all the historical brown baggers that we've had, but also we have a lot of fact sheets that are on the website, as well as the sire selection manual. So if you haven't been there yet, we would encourage you to go take a look. A lot of good, useful information for both you as trainers and also for your producers to send them to the website as well. So today with our session 3, we decided to go a little bit onto the environmental side of things, which as geneticists, we've always kind of shied away from just a little bit, but starting to take on a lot more prevalence here in what we do as geneticists, beef cattle geneticists. And so the overall theme for this session is going to be understanding methane from phenotyping to selection opportunities. And our first speaker for that today is Dr. Bailey Engle, and she's going to be talking about tools of new trade, methane phenotyping for genetic evaluations. And just to let you know, Dr. Bailey Engle grew up in a ranching and meat processing family from Montana. In her undergraduate education, she attended Montana State University in Bozeman. From there, she went to Texas A&M for her PhD in animal breeding, followed by a postdoc for applied cattle genomics at the University of Queensland in Brisbane, Australia. Bailey is now a research geneticist at the U.S. Meat Animal Research Center in Clay Center, Nebraska. She is part of the germplasm evaluation project team and is building a research program that in part focuses on developing genetic selection tools for novel phenotypes and challenging trait complexes. This includes recent initiatives to phenotype GPE cattle for both direct and proxy measures of enteric methane emissions. Dr. Bailey, Dr. Engle, it is a pleasure to have you on today, and we appreciate you joining us here for the Brown Bagger, and I will turn it over to you, and give me just 1 second to let you share, and then give me a moment and before you take control, and I'll mute myself and get out of here. So
sounds good.
Here we go.
All right, hopefully I have control now here pretty soon. Oh. I don't know what I'm looking at. There, I can still see your email. I don't know if everybody else can, but okay. Well, while we're getting set up, I was asked to talk about methods for phenotyping and how we can actually collect these methane phenotypes. So I'll be focusing pretty exclusively on that, and that I think will be a pretty nice introduction to what Troy will be discussing second. So just a quick little disclaimer, I will be referencing some specific product names and brands, and this does not represent an official endorsement from the USDA, but I'm just using it to give some specific examples, so please keep that in mind. Today's objectives are going to be pretty straightforward. I want to share with you guys the current approaches that we are having available to us for methane phenotyping. And then we'll briefly discuss the pros and cons of some of these approaches. And then finally, I want to share with you some of the emerging technologies, at least those that I'm aware of, in this space, because this is absolutely a growing field of interest. So let's dive right in. So the first things first, to get on the same page, what is enteric methane? Well, enteric methane refers to the methane that is produced as a byproduct of rumen fermentation, or fermentation in ruminants, I should say. It is the exclusive product of methanogens. That is the type of microbe in the rumen and the hindgut that produces methane. And the way that it does this is utilizing other byproducts from carbohydrate degradation. So some of these byproducts from carbohydrate degradation in the process of rumination include include hydrogens, CH4 or methane, CO2, carbon dioxide, or volatile fatty acids, or VFAs as we would often think about them. And so some of these important VFAs include acetate, propionate, and butyrate. And the reason why I mention these in particular is because increased levels of acetate and butyrate production correspond to increased production of methane, whereas propionate serves as a competitive utilizer of free-floating hydrogens and reduces the amount of CH4 that is produced through this process. And so this is really good for us, just as a high-level overview, because propionate is one of the primary precursors of glucose in ruminants. And so if the production of propionate can utilize some of those free-floating hydrogens, that not only increases energy availability to the animal, but also decreases methane production. And so really what I'm trying to demonstrate here is that methane is produced by the rumen microbiome or the hindgut, and it is a waste byproduct. CH4 eruction is associated with has been estimated to correspond to about 3 to 10% of energy loss from this process. So what we would like to do to not only reduce methane that is produced by each animal, but also we would like to increase the amount of propionate that these animals are producing. And one way that we might think about doing this is through genetic selection. But first steps first, we have to measure it. We have to measure how much methane each animal is producing. And that's what we'll be talking about today, mostly. So first, what makes a good phenotyping tool? Well, this is kind of my laundry list of what would be ideal. And very often there is not an ideal phenotyping approach or tool for any given trait, but this is what we would prefer in an ideal scenario. So we want them to be high accuracy, low cost, high throughput, where we can do a bunch of animals, non-invasive, and have low labor requirements. So this is a pretty tall order, but I want you to keep these kind of bullet points in mind as I discuss each of the potential phenotyping approaches, because these points will highlight where the pros and cons of each of these approaches kind of fall. So first things first is the respiration chamber. This is the tried and true approach for measuring gas output and input from animals. And so the way this works is that this is a whole animal open circuit kind of room that is used to measure respiratory exchange and gas exchange. And so what it's also trying to do is measure gas flux. And when I say gas flux, what I'm meaning is that circadian rhythm of gaseous output and oxygen input. And so that happens in a pattern throughout the day, and flux refers to the measurement of, or the representation of that entire pattern. And using respiration chambers with some pictures here, both a recent one from Cornell University that's recently gotten some press, as well as an example from Ilori in Kenya. The way that this works is that inflowing air is circulated around the chamber, and then it is pulled through the system and mixed with the emitted gases from that animal. And then the amount of gas that that animal emits is found by comparing the concentrations of those gases in the input versus those in the output. And so, as you can imagine, this captures all of the gas exchange from that animal during a given period of time, but it's also pretty labor intensive because as you can see in the pictures, it is a whole room. And so training those animals to go into the room can take some time. There's certainly high overheads and installation costs with these types of approaches, but it is really accurate. And so some alternatives have been developed to try to capture some of this accuracy, but with less overhead burden. So one of the alternatives to this is respiration head boxes. And so in this case, you can see that these plexiglass boxes entirely encompass and seal off the head of the animal and is able to then capture the methane that is either emitted via respiration or eruction. And there's an example here where you can see that the animal can lay down, stand up. But I think it should also demonstrate that there's a high level of animal training that is required and still a pretty high level of overhead costs. So another alternative to the standalone respiration chambers that has been developed largely in New Zealand are what they like to term portable accumulation chambers. And I almost said trailers because they have over time developed this methodology to be scalable, or at least attempt to be scalable, and have put these portable accumulation chambers, or basically little mini respiration chambers, on trailers to be able to take these trailers around from farm to farm to individually phenotype sheep. And in this scenario, it's a little different than the full respiration chambers, where the full respiration chambers, you typically would measure animals for, say, a 24-hour period. And with this scenario, they are measuring them for more like a 1 to 2 hour period and leveraging the high correlation between the respiration chambers and these accumulation chambers to demonstrate accuracy. They've also developed a similar design for cattle. And I haven't seen any results on these cattle portable accumulation chambers just yet, but I'll be really curious to see how it goes, in large part because they have shared that it doesn't require high training requirements like what I would associate with the head boxes or the respiration chambers. And I'm very curious to see how range cattle in the United States might do in one of these boxes. But regardless, it's another approach that is in line with that of the respiration chambers. So some of the pros for these approaches, and I've kind of generalized them all together because they are so similar, but there are some nuanced differences between them. But the biggest one is that the respiration chamber, particularly those whole room approaches, is the gold standard for accuracy when it comes to methane phenotyping. It is what we basically base most of our comparisons against, and it is our best approach for measuring methane emissions. In large part because it is a controlled environment where we have a lot of say over what happens in that scenario, and it is very good at measuring that flux, the pattern of respiration and gas emissions. And it is the respiration chamber is the only methodology that I'll discuss today that can measure both the ruminal and hindgut emissions. There's a little asterisk there because obviously the head boxes aren't measuring hindgut emissions. However, the cons are pretty large where it's the most expensive method. It's labor intensive, particularly because of the high training requirements for each of these animals to be able to go into the chambers or the head boxes, making it pretty low throughput. And one of the advantages of it is that it's a controlled environment, but it's also a disadvantage where we're not measuring the animals in their natural environment. And so we're making an assumption that by putting the animals into these respiration chambers, that is reflective of how they would be outside of the respiration chambers. And so to address some of these concerns, a second methodology was developed, and this is referred to as sulfur hexafluoride, or SF6 tracer techniques. So you might note from the graphic that I have there that this method has been around for quite some time. And what happens here is that a bolus containing SF6 is placed in the rumen of the animal, and this bolus slowly releases the SF6 gas at a known rate. And so then the animal wears a halter, as you can see in that picture on the left, that has a capillary tube that is connected to that halter, and then also a vacuumized vacuum container on their back, which you can see in the other little picture in the form of like a little backpack. And what this vacuum canister does is it pulls the air from that capillary tube, drawing in a sample of the gases that are respired or eructed from that animal. And so that would include then the CH4 that they have emitted, the SF6, as well as CO2, and then ambient air. And so then what CH4 or methane emission rate is computed relative to the known release rate of that SF6. So the SF6 is a tracer. And there's some real advantages to this, and the biggest one is that it allows for natural behavior to occur and for on-pasture measurement, which is in opposition to the respiration chambers. It's also a pretty accurate methodology, which is why it's been around and utilized for the last almost 30 years. But the downsides of this is that there is a high level of animal handling and labor required to train the animals to the halters and into the backpacks, to routinely switch out those canisters. And so there are pretty large training requirements, and there's quite a bit of literature out there discussing the challenges associated with this use that can really change the accuracy of the measurements that you're getting. And so there's some basically words of caution when utilizing this approach, but it's also one that can be very effective, particularly in research scenarios. And so those methodologies are geared towards trying to evaluate basically total emissions. And what we're moving into now are alternatives to that use spot sampling techniques to measure concentrations of methane from the breath of animals during short intervals. So this method is most commonly associated with the green feed measurement system. So this is a very specific company, but it is one of the only ones on the market that works in the way that it does, and it's the one that has been around for quite some time now that a lot of people trust. So the way that the green feed system works is it's a standalone automated head chamber system. And so it utilizes feed as a bait to draw the animal to it, to place their head into the air intake area. Then air is drawn past the animal's head and into the system while the animal is visiting the device, visiting the feed. And then the sample is then measured for gas concentrations. And so when we think about a green feed methane phenotype, what that actually represents is accumulation of many spot samples or many of those animals' visits. And if this happens enough times, which there are many people probably on this webinar who have worked very hard at determining how many samples we need. If this happens enough, it can be a good reflection of gas flux. So again, that circadian pattern of emissions. And that is obviously pretty advantageous. And one of the things that was really revolutionary about this when it first came out was that it was a really effective way to measure animals in pasture that didn't require the level of animal handling that was required by that SF6 approach. However, the downside to this is that it's quite expensive, both to purchase and then also the overhead cost of operating these devices or these systems is also quite expensive. And it does require some animal training where not every animal will train to the device, and you won't be able to collect a phenotype on every single animal exposed to the system. But very useful, and certainly the one that I think most of us on the call would be the most familiar with. And another alternative to this that utilizes this same spot sampling technique is termed sniffers. So this is a colloquial term, and all it refers to is that gas analyzer device, an example of which that is on the left. And so this methodology uses a continuous breath analysis in short durations from the expelled air of animals. So it is not so dissimilar to the green feeds in that regard. However, it uses infrared spectrum to identify the absorbance of the air sample in various gas densities to then determine gas concentrations from that continuous flow of the respired air. So there is an intake tube, which you can see in that picture, that is placed strategically near a spot where cows would be likely to place their heads. So there's some examples on the right-hand side of how this is currently being applied within dairy systems. So on the top is an automated milking system with a sniffer device applied. And then on the bottom, it shows some examples, again, from a dairy system of where this group placed their sniffer intake ports or intake tubes relative to a feeding bin. And so this methodology requires, again, spot sampling and repeated measures over long periods of time in a very passive way. But then it assumes that the precision of this approach is a little less because it is relying not on a calibration or relating to something like a tracer like SF6, but rather estimations of CH4 versus CO2. And so it is a little less accurate, but it is much more passive to the degree that many European dairies are starting to implement this in practice. And so the example on the left is from the Netherlands, where CRV, their primary dairy breeding company, is utilizing them within their barns. So the pros of this approach is that it is the most passive collection methodology that I'll describe today, making it pretty high throughput. The cons, though, is that it is lower accuracy compared to something especially like respiration chambers, but does have a pretty decent correlation to green feeds. And it is going to be a more sensitive approach to microenvironmental differences. And so by that, I mean the position of an animal's muzzle, the amount of airflow, the ambient air temperature, et cetera. It's more sensitive to some of those variables. And so then that means that it requires a very large number of spot samples in order to achieve a reasonable level of accuracy. And so this is good for scenarios where, like with the automated milking system, cattle are visiting basically the sniffer device for a set duration of time every single day. And it's also being applied more in a research setting to beef feeding systems where cattle are visiting bunks on a regular basis. So now as we start to get into some of the newer things, one I want to mention is the use of boluses to measure or estimate methane. So the one on the right is the only example I could find that actually had a infrared diode included on it that could directly measure gas concentrations. But the others that I have found, like those on the left that are currently being advertised as having capabilities of monitoring methane emissions, are actually using related traits like rumen pH, animal activity and rumination activity, I guess you could call it, and rumen temperature. So it's more of an indirect approach. And so I bring this up because there is quite a bit of attention being placed on the potential for using boluses for methane monitoring. But I think there are some real limitations to these approaches. But one of the pros is that once you administer the bolus, you have continuous monitoring and it's a one and done process. But that's also part of its downsides, which is once you buy the device, you only get to use it once on one animal. There's variable battery life, at least advertised amongst these three devices that I show here. And then most importantly, it relies on correlated indicators and prediction equations rather than any direct measurements of methane. And even if it did, it's one of the assumptions that it is making is that concentrations of methane in the rumen reflect that will be emitted. And that's a pretty strong assumption, but at the same time, it's not a one-to-one ratio. And so that is another limitation of this approach. And that kind of gets me into other proxy phenotypes. So those rumen boluses, especially the ones that aren't measuring methane directly, probably more like a proxy phenotype. And there are many others as well. So one of them that's getting quite a bit of attention is the use of the rumen metagenome and a profile of that metagenome. So if we recall back to the beginning, methane is the product of microbial fermentation, specifically methanogens, but the composition of that entire rumen microbiome will determine the amount of propionate, the butyrate-propionate ratio, and the number of free-floating hydrogens that are available to the methanogens to make methane. And so the idea is that if we can sequence and capture the diversity and the abundance of these various microbial species, that can be an indicator of CH4 production. And it has been shown to be a highly correlated indicator and is therefore has potential to serve as a good potential proxy for methane production. Another one is that's a little less relevant to the beef side, but very, very relevant to the dairy world is milk mid-infrared spectroscopy, or MIR. So the milk MIR reflects the composition of that milk, and it is a process that is routinely applied to both bulk milk samples and individual cow milk samples. And it is utilized in routine practice to determine things like milk payment and milk records. So this is being evaluated on majority of dairy cows as it is. And it has been demonstrated to be able to predict methane emissions, in large part because the composition of the milk, particularly the lactose, fat, and protein compositions, are directly related to volatile fatty acid production in the rumen, which, as we know, volatile fatty acid production in the rumen is also directly related to the amount of methane that is emitted. And so because of that interrelatedness between these processes, the infrared spectral profiles can be predictive of methane as well. And this is something that is being heavily explored in the dairy industry. And then one that's a little bit more relevant to our beef cattle world is feed efficiency, where studies have shown already that selection for lower RFI or residual feed intake or more efficient cattle can reduce CH4 production. And this, I think, makes a lot of sense because more efficient animals with more efficient rumens are going to produce less methane, and they're also hopefully going to be more efficient in their growth. This is useful to us because we've already been collecting quite a bit of feed efficiency data over especially the last 15 years. And even though this is a pretty expensive phenotyping approach, it's something we already have available to us. And the simultaneous collection of methane and feed efficiency data is very, very advantageous. There's also quite a bit of work being done to improve our ability to effectively and efficiently collect feed efficiency information. And so I include the example of a camera imaging system across the bottom. And this is a system that is currently being applied in Denmark within Viking Genetics dairy farms. So that's the primary dairy breeding company for Denmark. So proxy phenotypes, hopefully, they are more easily accessible, and that's why we care about them. They're often more cost-effective, and hopefully, data collection can already be something that is fairly routine. But the downsides to using proxy phenotypes is that we still need to relate it back to direct methane phenotypes. So we still need to be collecting methane information. And there's also variable correlations, especially genetic correlations with actual methane production. And so this can reduce the accuracy of selection utilizing these approaches. But it's something that we should absolutely be thinking about if we wanted to perform genetic selection for reduced methane, and a really useful tool. So very quickly, because I'm running out of time, I think I might be going over. There's a couple examples of new and emerging tech. So this is a company based out of the United Kingdom, and you can see here their current rendering of their device, which is basically a very miniaturized sniffer that sits right there on the bridge of the nose of these animals as part of an integrated halter system. It uses accelerometer data and information about respiration rate to then be able to estimate as well as direct collection, of methane, but to then estimate a emissions rate for CH4. And then one of my other examples is a company or a joint effort between two companies out of Australia called Optaway and Accent. And so Optaway builds these front-end weigh devices so the animals that visit the basically the mineral lick that they're using as a bit of a bait or incentive to visit the device have a front end weight taken. And then they've integrated a sniffer device as well so that there's simultaneous measurement of both weight and methane production or emissions. So in summary, and I'll keep this part very brief, there are quite a few different approaches available to us, and each of them have their pros and cons. And as a scientific community, we have not reached a consensus as to which way is absolutely best. And I suspect that that is because we don't know that yet, which way will ultimately be best. As demand increases for these types of phenotyping approaches and phenotyping devices increases, I would expect that there will be more approaches developed. I am aware of other companies that have been working on similar efforts, some of which I was not given permission to talk about today. But for those of you who are interested in methane and methane phenotyping, definitely keep your eye out next year because I would expect there to be multiple announcements from some companies that you would be very familiar with. So with that, I will turn it back over to Darrh and Troy, and thank you guys for your attention.
This control thing drives me crazy. Anyway, I'm back in control. What was happening before, Bailey, is we were fighting each other. I was trying to get over to mute myself, and you were trying to take over control, and we kept stealing
Sorry
it from
about
each
that.
other. So we're good. So if anybody has any questions, please put them in the question and answer part. It looks like we have one from JR. He asked, how long in days do you need to collect methane production to represent the animal's overall methane production?
So if you were using a respiration chamber, generally speaking, they have found that 24 hours worth of data collection is sufficient for that. And same with head boxes. The portable accumulation chambers, they're measuring those animals for 1 to 2 hours at a time, and they have found that that is effective at getting a decent phenotype. But from the spot sampling techniques, there's a bit of a difference between how many visits or how many measurements would be required to get a good methane phenotype versus some estimation of overall production. So where I'm trying to draw the line is that we can get a good representation from a green feed device after about 40 visits, I think is what people have found. If you are very interested, Kansas State and specifically Elizabeth Dressler has done quite a bit of work looking at this exact question. But to get more of a overall, I guess, definitive production, like how much methane are they truly producing, I think it would probably take a bit more, but that's a slightly different question than what geneticists typically ask, so I'm not as sure.
Excellent. All right. If you have any other questions for Bailey, just put them in the question and answer. And Bailey, if you're going to be able to hold on with us, we'll go ahead and get Troy going, and then we can have a question and answer session at the end. All right. Next up, we have Dr. Troy Rowan at the University of Tennessee is going to be talking about selecting for methane emissions, global examples and opportunities in the U.S. beef industry. Troy was a bad boy and didn't send me his bio, so he gets to hear my version of his life. Dr. Troy Rowan, now here's the one I think grew up in Illinois. Is that correct?
No, wrong I state there. I'm from Iowa. Yeah.
Okay, sorry. But anyway, he's the only Creighton Blue Jay that I know, I think. So and then got his that's where he went undergraduate and then got his I think both master's and PhD at Missouri.
Yes, sir.
Okay, and now at the University of Tennessee, and being my partner right across the line, Troy and I have had the opportunity to work a lot together, and really appreciate what Troy's doing down there, and look for good things out of Troy in the future. And with that, Troy, I'm going to turn it over to you. Now, give me a moment. Wait till you see my picture disappear before you try and take control, so that we don't get in a scrap here.
All right, I'll
Okay.
hold off from doing any clicking. But thanks, Darrh, and a big thanks to Daly there. That's going to save me a fair bit of time. I'm at BIF board meetings in Kansas City and have a flight to catch. And I thought there was no chance that I would ever get through my slides. But Bailey gave a really, really good rundown on the pluses and minuses of each of those phenotyping strategies. You'll see a lot of that pops up over the course of my presentation. But just a little bit of background before I start into the presentation. Let me see if I can get this to work. Maybe. There we go. All right. So I guess just a little bit of an overview of where I'm going to go. I want to both touch on the context of this larger question of methane emissions. So why do we care about it? What role does genetics have to play? I think that there's a lot of work around the world in particular, and I wanted to give some global examples today. Their context is different than in the United States. So trying to understand those differences in context, why our friends in New Zealand, Australia, the UK, Ireland, Canada have maybe been a little bit more ahead of the curve on some of these genetic approaches to curbing methane. And then talking about a couple of practical examples where estimated breeding values are being calculated. Producers have tools that they're actively using to reduce methane emissions from their cattle and other ruminants. So with that, I want to jump in and just give a really broad overview from my perspective on where genetics and methane sort of overlap. I've been pulled into more and more conversations in the last couple of years surrounding this methane question, and oftentimes there's one geneticist sitting around and there's 10 different people with some sort of a feed intervention that reduces methane, and we're trying to figure out, is it one or the other? I think that the consensus that we've settled on is that it's both, right? Genetics takes a long time, right? Our generation interval in cattle is 5 years on average, say. So making genetic selection decisions, that's a long-term game. Many of these feed interventions, dietary interventions, management interventions, those are immediate changes in that individual animal's methane emission. The flip side of that, and where I think genetics fits in alongside these interventions, is that genetics is cumulative, right? If we make genetic progress on these traits, we don't lose that the next generation. We don't lose that the next year. If we stop feeding a feed additive, the effects on methane emission are going to go away whenever we halt feeding that additive, right? So genetics are cumulative, they're constant, they're gradual, and most importantly to the beef industry, they're able to happen in these extensive systems, right? Delivering a feed additive in a dairy is very straightforward. Doing it in a feed and a feedlot is very straightforward, but delivering some sort of a feed intervention to cows in extensive rangelands is very, very hard, right? So genetics is one of our ways at chipping away at the grazing cow, which overwhelmingly makes up the United States population of ruminants, right? How do we chip away at the methane question? Genetics is a big piece of that, right? But as we all know on here, our ability to make genetic progress is going to be capped by one, measuring phenotypes, and two, delivering genetic predictions, right? So this is familiar to everybody on the call, right? But regardless of what our trait is, if it's heritable, and hopefully it's economically relevant, which we'll talk a little bit more about here in a second. If it's heritable, we can measure it. We can generate genetic predictions, and with those predictions, we can accelerate our genetic progress, right? We make the right decision more often than not, and that's really going to help us move the needle on a trait sooner rather than later. The flip side of this, right, is just what Bailey talked about. Phenotypes are going to be our limiting factor. We've got millions of genotypes in some cases in beef breed associations. This is from the American Angus Association a couple of years ago. We're genotyping our cattle at a massive scale. And what that means from a novel trait development standpoint is that with a concentrated effort on phenotyping the right animals with the right strategy, we're able to develop these genetic selection tools that are quite accurate. Genomics just helps us make those more accurate for our young, unproven animals that are going to become those future parents in our population. So on a more, I guess, quantitative genetics note, right, the big picture, before we start developing tools to select for a trait, we want to know that the trait is heritable, right? And it turns out that methane production is heritable in beef cattle, dairy cattle, sheep, all ruminants. Elizabeth Dressler, Bailey mentioned her in her presentation, has written a really nice review paper here talking about some of these heritabilities, pulling in data from a number of different studies over the past decade or so. And these heritabilities tend to be somewhere in the 0.2 to 0.3 range, right? These are very similar to a trait like weaning weight. So there's additive genetics that we're able to select for and move the needle on a trait like methane. And the other thing that I think that we are working on making the case for right now is that our continued genetic progress across efficiency traits is helping us when we think about methane intensity. And that's a concept I'll get to in a slide or two, right? But the thought being that raw methane emissions aren't particularly useful unless we're scaling them by some sort of an end product, right? So in this case, right, growth and productivity, and in particular, our forage-based cow-centric traits, think fertility, think cow longevity. These all help increase the overall efficiency of our product. And so as we think about being sustainable, right, doing more with less, these growth, productivity, cow-centric traits are all very, very important components of that as we scale things like methane by that overall animal productivity. So that's the good news that we have a good handle on. We're not going to stop making genetic progress on these traits that are a part of our current genetic evaluations, indexes, and our breeding goals as a population. But there are another set of bigger questions, I think, as we dive into this methane world that we still don't have a great handle on. They're going to be really, really important over the coming years to figure out, right? And I think generally we still have some outstanding questions on what the genetic correlations, the pushes and pulls, the antagonisms possibly that exist between selecting for methane and selecting for some of these other traits that we know are economically and in many cases sustainability relevant, right? It's pretty clear that there's a direct relationship between dry matter intake and methane production. We know that larger animals tend to make more methane generally. Dave Wallman's group at Oklahoma State has done some really nice work on this that says, maybe that's true on the aggregate, but certainly some corner cases where that's not always the case. And Drew LeCamp from University of Nebraska has written a really nice paper exploring both phenotypic and genetic correlations between methane emission and a variety of other traits. And the conclusion that we've sort of come to on feed efficiency is that there's probably a positive relationship or a beneficial relationship, right? More feed efficient animals maybe tend to be lower emitting animals, but that's still a little bit fuzzy in the big picture. And I think a bigger question that we have to ask as an industry, as a seed stock industry in particular, before we start making these breeding decisions, understanding what the market drivers are in the space, right? That's really going to dictate how we integrate things like methane into an overall breeding objective or into an index, right? Understanding how a producer gets compensated for collecting these phenotypes, for integrating selection against methane production, possibly into their breeding objective, is going to be a really, really big moving piece that I think is still pretty fuzzy now, as carbon markets they remain the Wild West as far as I'm concerned. Understanding whether these are going to come as insets, so a supply chain demanding this, or offsets where we sell a reduction in methane in our herd to another entity who's a carbon contributor, right? So again, lots of questions. We could do a whole talk about this, and there are many, many people that have a better handle on how the marketplace is going to work out. I'm here mostly to talk about the genetics and how we might implement these things. And one of the things, whether it's feed efficiency or methane emission, I think we still don't have a great handle on whether or not we should treat feed efficiency or methane emission on concentrate as a separate trait from feed efficiency or methane emission for grazing animals, right? Has really big implications for how we measure these, how we undertake phenotyping efforts, knowing that the vast majority of our emissions come from those grazing cows. That has some pretty big implications on whether we test them in a dry lot as heifers or whether there's real value to measuring these out in the field in those more extensive situations like Bailey talked about in our mature cows. And so our challenges, right, and Bailey did a really good job underlining this, I won't go in too much more depth, right, is that phenotypic collection right now, I've been at the BIF board meetings all week. We've been talking about how do we incentivize phenotype collection, how do we fill in some of those missing points in our genetic evaluations, and this is not a straightforward concept, right? But the challenge with methane, right, is with no clear market signal, right, no clear value that we're able to place on these traits themselves, and with these traits being very expensive to collect, there's limited data sharing even amongst academic groups, and producers certainly were not in the realm of possibility for widespread phenotypic collection. I did some rough calculations of a green feed phenotype. This is from one of our research centers, and I figured that a somewhat reliable phenotype, as sort of outlined by that of the paper by Dressler and colleagues, when I sort of depreciate out my green feed, I factor in some of our research farm labor, the time that it takes to get this, the usage of the green feed by cows. This is somewhere in the neighborhood of a $75 to $100 phenotype to collect. Right. This is very expensive in the immediate term. Lots of exciting developments to keep chipping away at this, but I'll just underline this, that at the current moment, our gold standard for measuring these things in an extensive system is very, very expensive to accumulate these phenotypes. Certainly nothing that we can expect producers to pick up in the near future. So the sort of bit that I'll add here, right, is a little context on how we define traits, right? So understanding that the trait that we're measuring is methane emitted per day typically. Again, whether that's in a concentration, whether that's a gas flux, right? The way that we can scale that really determines how we think about it and would implement this in a genetic evaluation, right? So I talked about methane intensity. We can think about methane intensity from the perspective of live weight of an animal. We can think about methane intensity in the context of kilograms of beef produced at the end of the day. We can scale methane production by dry matter intake, right? So we've got another piece of information, calculate methane per unit of dry matter consumed, or we can factor in all of these pieces, right? Live weight, dry matter intake and live weight, and get some sort of a residual methane emission to compare between animals, right? I put this up here, one, because I think it's a really good illustration of these different trait definitions, but also trying to understand, again, the application of a prediction for methane is going to be very different depending on the entity that's interested in it, right? If we're trying to underwrite carbon credits on a farm, we're going to be looking at something different than if we're a supply chain who's maybe interested in this methane scaled by the units of end product, right? Lots of moving pieces here that I won't get into too much more detail on. So the thing I'll add a little bit more context on as well is why are other countries ahead of us, right? We've had lots of this work done, particularly in New Zealand. These are our leaders in this space. And the context that's really important is to look at the proportional amount of greenhouse gas emissions on a country-by-country level, right? New Zealand, a small country, relatively small population, but a fairly large and vibrant agricultural sector. Tons of grazing, right? Anybody who's been in or around New Zealand knows that it's one of the greenest places on earth, right? Great for growing grass. And as such, nearly 50% of the country's overall emissions are going to come from the livestock sector, overwhelmingly, that proportion coming from dairy cattle as well as beef and sheep, right? So compare that to the United States, where our livestock industry is making up somewhere in the neighborhood of 2 to 3% of our total greenhouse gas footprint, right? The priorities are shifted, the pressure from governments, from supply chains, from consumers, as countries look to meet the Paris Climate Agreement, the various agreements that come out of COP, is shrinking their environmental footprints the place that has the most fat on the meat in New Zealand very clearly in agriculture there. So they've been forced to make this a major priority in how they handle this at the country by country level. And so I'll give just a couple examples, one from New Zealand, one from Canada, on how they're making these genetic evaluations work. The first, I think Bailey did a really good job describing the toolbox that the folks at Ag Research, which should be a government-funded research organization in New Zealand, they've really taken a brute force phenotyping approach to this, right? They've built these awesome little portable accumulation chambers. They've phenotyped thousands of sheep and now have a genetic evaluation that's online for their subset of their producers that have undertaken these efforts on the phenotyping space. And I think that this is a really interesting one, and one that has a lot of appeal to the beef industry, right? Can we bring these things on farm, get a one and done phenotyping done? This is just like bringing your cattle through to get them ultrasounded, right? We've got a professional there for a full day. It takes a little time, but when we're done with it, the phenotyping is over. We're not having to maintain a machine. We're not having to worry about partial usage, right? Everybody gets a phenotype, and we can knock it out over the course of a couple days. So again, excited to see the potential of these portable accumulation chambers possibly serving as genetic tools for us in the beef industry. One of the other interesting things that has come out of the group at Ag Research is a long-term selection experiment, single trait selecting for methane and trying to understand how that affects other correlating traits. One of the things that I think is very interesting is that they've found not any major impacts on productivity, right? So lamb and ewe methane emission is highly correlated. So measure on a young animal is probably going to be predictive of the measurements on an older animal out in a pasture-based setting. Methane emission phenotypes are really important in pasture settings, obviously. So understanding their impacts on things like fertility and productivity of those animals is quite important, right? So in the absence of any changes to fertility, to feed efficiency, meat quality, or carcass traits, it looks like methane can be one of those traits that fits into a breeding objective without getting us too far off track of the overall profit-aligned breeding goal in many of these cases. The second example that I'll give is from the Canadian dairy industry. And this is a bigger challenge, right? Cows are trickier to phenotype than sheep that you can stick in a box, 12 sheep at a time in New Zealand. So this is work that was done by Lactanet and the University of Guelph, doing large-scale phenotyping efforts in Canada and delivering the world's first cattle-focused methane prediction. So the challenge for us as an industry and people that think about measuring phenotypes at a high rate, right, enough for genetic evaluations. What do we do when it's too expensive, right? We find an indicator phenotype. And I'm really glad that Bailey introduced this idea of mid-infrared spectroscopy, a great tool that the dairy industry has. But effectively what the Resilient Dairy Project, this was centered at the University of Guelph, Dr. Christine Baez's lab, in partnership with Lactanet, they gather a large number of green feed phenotypes. I think there are well over 1,000 cows now that have been phenotyped, and trained machine learning models to pick out these mid-infrared spectroscopy patterns that match up with those animals' actual emissions as measured by a green feed. And then, since this is done routinely on very large numbers of cattle, they're able to turn this into a genetic evaluation based on these MIR, these mid-infrared spectroscopy methane values, right? And so this predictions for methane produced by a first lactation Holstein, sort of 120 days plus in milk, right? And the beneficial thing here, a genetic evaluation can't happen on 1,000 records, but it certainly can on 700,000 records, right? So these MIR records allowed this to get turned around. Certainly not perfect, right? Our indicator traits, we want them to be as close to the trait that we're interested in as possible. But this really accelerated the delivery of the tool, and I know that they're continuing to explore other areas where they can continue to collect larger numbers of traits that are a little bit closer to the real phenotype itself, including putting a large number of sniffers in AMS systems across the country But again, this is a good example of a tool that being developed in really short order in the context of a phenotypic collection that balances those more expensive phenotypes with a cheap, easily accessible indicator trait. And then ongoing developments, I mentioned this, putting sniffers in AMS systems has become a commonplace in the dairy industry in Europe, Canada, and New Zealand. And one of the things that I know that they're thinking about, and we as a genetic evaluation community should be thinking about, is how do we model these different sources of phenotypes, right? A combination of respiration chamber, which is really the gold standard, right? Some portable accumulation chamber data, some green feed data, some sniffer data. Modeling those jointly is probably going to be the approach that we have to take to leverage all of those, get the largest number of phenotyped animals, understanding that those traits will act a little bit differently, have different sources of variation that we need to adjust for, but no reason that we can't utilize those in concert with one another to strengthen our genetic evaluations. And so, again, the beef industry has a number of challenges, right? Cost prohibitions, right? The collection and extension systems. How do we data share these limited phenotypes? Are they going to live in individual breed associations? Are they going to live in individual academic groups? I think we still need to figure that out a little bit. But in particular, when we think about genetic evaluation, how we handle data heterogeneity and quality, right? Some cows that we have on our green feeds have visited hundreds or even thousands of times over the course of, a handful of months. Other animals have only visited, 3 or 4 times, right? But there's possibly still some valuable information in those animals that have not gathered a complete methane phenotype. So we need to understand how we might utilize those, and then getting a handle on the market signals and how we fit these into an index is going to be of great importance. And so the last thing I'll mention is this idea of the microbiome, not even so much as an indicator. Bailey talked about that, and it absolutely has a potential there. But even when we have a good methane record, can we move a little bit more towards phenotypic prediction when we have an understanding of the animal's genetics as well as of its rumen microbiome, understanding that animal genetics is both going to impact the animal's actual methane production as well as the production of the methanogens that thrive in this rumen microenvironment. And so the group at Ag Research in New Zealand has done some really interesting work where they jointly model both genetics and these animals' microbiomes in their genetic prediction equations, allows them to nearly double the prediction accuracy for something like methane production in these animals compared to just using a genetic model. Also, I think there's a lot of value in the microbiome allowing us to account for environmental variation and move towards this prediction of phenotype, right? The forage that animal eats is going to drive changes in that microbiome environment. Can we reflect that and better capture the phenotypes that we care about? And so with that, I think I'm going to just skip to the end here. I don't have time to talk about ongoing projects in our lab, but this is something that we're working to find some of those indicators, those approaches for measuring the microbiome in a lower input, more easily attainable manner. And so again, I know there are a number of groups, many of them on this call, that are working in the same direction. So an exciting space to be in, and really I think is a research community trying to stay ahead of the game, right? Being able to have tools that we can roll out in the near future so that we're staying ahead of whether it be government regulation, demands from our consumers, but making sure that the beef industry is able to sort of agilely deal with this as it rolls out. So with that, some takeaways, my contact information, and happy to answer any questions if we've got them here.
All right. Thanks a bunch, Troy. That was very enlightening. And if you have any questions, everybody please put them in the question and answer box. And that can be for either Dr. Engle or Dr. Rowan. Looks like we have a question. So which of these methods might be influenced by CH4 from bedding? Oh, okay.
I think this might be a question for me. So of the methods that I described, there's really not very many that would be likely to be impacted by methane from bedding. One would be respiration chambers. Like if you were to include bedding into that, it would be accounted for in those measurements. Most often though, we don't include bedding in the respiration chambers. The other one though would be the SF6 tracer approach, particularly and maybe even exclusively if you were to use that approach indoors, which is not really recommended because of potential for interference, both with the SF6 accumulating within the building, as well as potential for things like environmental uh, emissions to be accounted for in that same way. So those are kind of the two ways to think that you would maybe need to be worried about this, but otherwise not too much of a concern.
All right, thank you. I actually got a message from Troy. He's obviously concerned about getting home tonight. He had to take off for the airport. So Bailey, you get to answer all the questions now. So if anybody has any other questions, please put them in there. I have one for you, Bailey, that came up
Sure.
as kind of a concern, I guess, and it has to do with a little bit of what Troy talked about in terms of economic relevance of the trait and all. And I think we all know the big picture importance
Mm.
and to the industry, to the world, everything. But I think of a trait like tenderness. You know, we've had tenderness as a measurable trait for a long time, you know, highly heritable and everything else. But, you know, that's one that we've always had such a hard time pinpointing, you know, the value of it to the consumers. We know there's a value, but capturing that value has been difficult. So we haven't made as much progress there as probably we could have or should have or things like that. Any thoughts on that? I mean, because we really are going to have to do a better job, I think, with this one of getting that message out there and trying to really show that value.
I think about it like tenderness also for the same reasons, where currently, at least in our country, there is not an economic incentive to select for reduced methane emissions. At least from our livestock, there is not. And I don't know exactly what that incentive might look like. Troy kind of described the ways that it could work. But the things that we have seen in other countries, which is what I would expect would be the most likely outcome here, is modeled after something that happens other places, is that a lot of this is consumer driven and driven by consumer preferences and demands. And then sometimes there have been government interventions or taxes placed on something like methane production from each individual animal. And that is something that is pretty scary for us to all to think about, but I think is probably the least likely outcome. I think most likely that if there are incentives placed on reducing methane, that it will come from consumers. But I don't know how we incentivize it. At this point in time, I don't necessarily have an expectation for producers to immediately start phenotyping methane extensively or utilizing these selection tools. But I do hope that as a research community that we can develop enough resources so that in the event that incentivization does happen, that we are prepared and ready, in particular so that we can maintain our market share, maintain consumer demand, et cetera. But this is a process and we don't necessarily know where it's going to go. And I don't have a great prediction for that at this point in time, but I am involved because I want to be ready if that eventuates.
Yeah, I think that's great. And I think we also need to not be shy about getting that point across that I think both of you may have mentioned that what we've done from a production side of thing, improving the efficiency. I mean, whenever we can improve reproductive efficiency, that does a lot in
Yeah,
terms
that's the one
of
thing.
go ahead.
Sorry, go. I was just going to say that is one of the things that I think is really an advantage of methane, or thinking about selecting for reduced methane, is that we're also simultaneously selecting for improved efficiency, or at least we have the opportunity to do those two things simultaneously. It's a pretty expensive way of thinking about efficiency. Like these methane phenotypes are expensive, but at least if we are selecting for reduced methane, we are also trying to improve the efficiency of our cattle systems.
Yeah, and I think the inverse as well, that as we do
Mhm.
those other things, that we're in effect impacting our methane production as well.
For sure.
Any other questions, folks? Just pop them to the Q&A. If you have any lingering questions, probably the best route is either directly contact one of the speakers, or you could also send it through Dr. Bob Weber at K-State, and he can disseminate that to the speakers and in a forum format. So if you have anything that comes to mind, we'd love to keep this discussion going. I would like to thank again our speakers for today, Dr. Bailey Engle and Dr. Troy Rowan, and remind everybody that session 4 is next week. It's going to be beef industry research innovations. We have Dr. Mark Bogus from Meat Animal Research Center. Have a lot of representation, I guess, from you guys, and we do appreciate all that you guys do at the Meat Animal Research Center. And also Dr. Dwayne Wolf is going to talk about beef yield grading, history, issues, and opportunities. You will also get your invitation to next year's BIF symposium, which is going to be in Texas and going to be hosted by the West Texas crew. So we appreciate them as well, and hope everybody will join us next week. I did forget to mention my cohorts in this. We have Dr. Bob Weber and Dr. Matt Spangler. Sure appreciate those guys. We work as a committee to put this together. I think we're in our 21st year of doing this now, so it's been around for a while. Great to see the attendance, you guys that show up every week. And so we will see you next week for the last one. And with that, we'll sign off and say, everybody take care and be safe.